Hello, dear students. Today we're going to speak about DNA structure. Let's start. The description of the double helical structure of DNA by Watson and Crick in 1953 was a landmark in science history. Their discovery sparked off a new era in scientific research which has had and will continue to have far-reaching consequences. A polymer of nucleotides. Each DNA strand is a polymer made up of nucleotide subunits. The nucleotides join together to form long, unbranched polynucleotide chains. Each nucleotide consists of deoxyribose, a 5-carbon or pentose sugar, an organic nitrogen-containing base, of which there are 4 different types, and phosphoric acid. The sugar and the organic base join together by a condensation reaction to form a nucleoside. A condensation reaction results in the removal of a water molecule. Another condensation reaction joins the nucleoside with phosphoric acid to form the nucleotide. This bond forms between carbon 5 of the sugar and the phosphate and is called phosphoester bond. The organic bases present in DNA are either purines or pyrimidines. Purines have a double ring structure. Pyrimidines have a single ring structure. 2 nucleotides can join together by a condensation reaction between the phosphate group of one nucleotide and the hydroxyl group on carbon 3 of the sugar of the other nucleotide. The bonds linking the nucleotides together are strong, covalent phosphodiester bonds. The process can be repeated so that the polynucleotide chain builds up. The chain has a sugar-phosphate backbone with the organic bases projecting outwards. Each chain has 2 distinct ends, a 3' end and a 5' end. At the 3' end, the carbon 3 of the deoxyribose is close to the end. At the 5' end, the carbon 5 of the deoxyribose is closest to the end. The double helix. DNA consists of 2 polynucleotide chains coiled around each other to form a double helix. The double helix is held together by hydrogen bonds between pairs of bases in the 2 chains. The pairings depend on the shapes of the bases. A purine can only bond with pyrimidine, and on their ability to form hydrogen bonds. Adenine pairs with thymine, forming 2 hydrogen bonds. Guanine pairs with cytosine, forming 3 hydrogen bonds. Complementary base pairing. These complementary base pairs are the only ways the bases can bond and join the 2 nucleotide chains. Thus, the sequence of bases along one polynucleotide chain determines the sequence along the other. An adenine on one chain means there must be a thymine on the other chain at that point, and so on. Complementary base pairing forms the basis of DNA replication and its ability to form messenger RNA during protein synthesis. Complementary base pairing can happen only if the two polynucleotide chains are antiparallel. Antiparallel chains run in opposite directions. One chain runs from 3' to 5', and the other from 5' to 3'. Watson and Crick's model of DNA showed that the base pairs are 0.34 nanometer apart and that each complete turn of the helix has 10 base pairs. In summary, DNA is a double helix made of two polynucleotide chains. Each chain has a sugar phosphate backbone on the outside with organic bases on the inside. The two chains are held together by complementary base pairing. The chains are antiparallel. The 5' end of one chain lies next to the 3' end of the other chain. That is the end of our video session. Next time we're going to speak about chemical nature of genes. Thank you for watching.